Hello and welcome to IB Biology Notes with Dr. Kimmo. This is B1.2 proteins. This video is about amino acids, peptides, and of course proteins. In higher level, it's also about the structure of proteins. Proteins consist of monomers called amino acids. Each amino acid has an alpha carbon, and that alpha carbon binds an amine group, a carboxyl group, a hydrogen atom, and a side chain called the R group. The amine group is basic and the carboxyl group is acidic. That means that the amino acid is amphiprotic. It can act both as a base and as an acid. The R group can be many different things. Two amino acids can be linked together to form a dipeptide. This is a condensation reaction between a carboxyl group and an amine group. Water is released and the resulting bond is called a peptide bond. Two or more linked amino acids form a polypeptide. Shorter polypeptides are also called oligopeptides. Then you can talk about dipeptides if there's only two amino acids, and tripeptides if there's three amino acids. Polypeptides are the main component of proteins. We obtain amino acids from food, and our ribosomes make polypeptides from 20 different amino acids. Each amino acid differs by its R group, and that's illustrated on the right, where all 20 amino acids can be found. You can see that each of them has the same backbone with an amine group and a carboxyl group, and then they have their own type of side chain of R group. Plants can make their own amino acids through photosynthesis, but animals obtain amino acids from food. Humans can synthesize 11 of these amino acids from other amino acids by modification of the R group, and these are called non-essential amino acids. The rest, 9 of them, are essential amino acids, which means that we cannot synthesize them and we need them from food. The essential ones can be seen here in yellow, and obviously you are not expected to know details like this. Phenylketonuria is a condition associated with the synthesis of amino acids. Most people can synthesize the amino acid tyrosine from the amino acid phenylalanine. This is done by an enzyme called phenylalanine hydroxylase, PAH. However, people with a rare genetic mutation in the PAH gene cannot synthesize tyrosine, and they have phenylketonuria, PKU. What this leads to is that phenylalanine and its metabolites accumulate and can cause severe mental retardation. Also, tyrosine becomes an essential amino acid. However, newborns are routinely screened with a blood test, and there is effective treatment. That is, diet very low in protein throughout life to keep the phenylalanine at an acceptable level. Also, aspartame is to be completely avoided because aspartame is broken down to phenylalanine in the body. Aspartame is an artificial sweetener which can be found in things like soda, candy, diet foods. There is also a medicine which enhances the function of the enzyme PAH. But for this to work, the PAH enzyme needs to have some functionality left. The number of different polypeptide chains that a cell can make is essentially unlimited. Let's do some math. There are 20 amino acids, and if you want to make a dipeptide, you have 20 times 20 different alternatives. That's 400 different alternatives. If you want to make a tripeptide, the number is 20 times 20 times 20. That's 8,000 different alternatives. A polypeptide with 100 amino acids, there the number of alternatives is larger than the number of atoms in the observable universe. Yet many polypeptides are much longer than 100 amino acids. The longest human polypeptide is titin, present in muscle with more than 34,000 amino acids. The illustration shows alpha amylase, which is present in saliva. It consists of a polypeptide with close to 500 amino acids. Proteins are sensitive to temperature and pH. Proteins form 3D structures through bonds or interactions between R groups. If you're doing higher level biology, this is something you will be learning more about later in this video. The 3D structure is essential for protein function, and the bonds and interactions are generally weak and sensitive to changes in temperature or pH. Denaturation is when a protein loses its 3D structure, and that's what happens if you, for example, fry an egg. Then the water-soluble proteins in the egg are heated and denatured. They lose their 3D structure and they become insoluble in water. Denaturation is usually irreversible. That's all for now. See you next time.